Hi students, welcome to HSC Biology and Module 5, Heredity. This is video number 10, and in this video we're going to be concentrating on meiosis. Just as we did previously on the last video, this is also about modeling processes involved in cell replication, but this time we're going to focus on the processes involved in meiosis. So again, what we want you to do is to try and tick off these levels when you feel like you've achieved them. So firstly, just being able to suggest why reproductive cells have less genetic material inside them. So understanding the reasons for the reduction in genetic material for sexual reproduction is an important starting point. To be able then to describe the stages of meiosis, again, in general, I guess, is where we start, and then we try and get to some more of the specifics. And in this case, what we ideally want you to be able to do is not just to model the processes involved, but also to include one very important consequence of the process of meiosis and the increase in variation that it produces, which is a process known as crossing over. So we're going to talk about that and also all of the key steps involved in meiosis in this video. So by way of introduction, let's just compare mitosis and meiosis before we get too much into this process. And as we've talked about previously, when you've got two processes like this that are quite similar, drawing a comparison table can be really helpful to identify some of the important features of each of these processes. They're both cell division processes, but there are differences between them. And either using mitosis, meiosis, similarities and differences, similar to this, mitosis, meiosis, sim and diff. And this sort of thing is a good way of giving you a nice summary table. In fact, you can even remove the line between the similarities, because obviously similarities are similarities, they're common to both. And it allows you to identify some really key differences between them, but also the fact that they do share some characteristics in common. So that's an opportunity for you to do. You can actually do that while you're watching this video, or particularly if you've watched the previous one on mitosis, or you can have a look at it in your textbook, or you can look at plenty of resources online in which this has already been done multiple times previously. So a couple of differences. Firstly, in mitosis, we have a single division process. In meiosis, that division is a double cell division. Now, the first step is a replication. So that is common to both processes. But in meiosis, we go straight from the first division, the cells splitting into two, into a second division without the replication stage. And this is one of the reasons why the number of chromosomes is actually reduced in order to produce cells with only half the number of chromosomes as the original parent cell. There's a specific to the gonads, and it is for production of gametes. And in this particular video, we will also look at the differences between oogenesis, the process of producing eggs, and spermatogenesis, the process of producing sperm, because there's a few additional differences associated with meiosis for those two different purposes. The genetic material is replicated, and that does happen just as it does for mitosis, but this time we have two division sequences, which means our diploid number of 2N chromosomes is actually going to be reduced to just half that, just N numbers of chromosomes in the gametes. And the other important process is that in mitosis we have one cell splitting into two identical cells. In meiosis we have that same beginning, but then each of those splits into two as well. So ultimately we end up with 4 in the process of meiosis, whereas we only have 2 in the process of mitosis. And so each of these will have the same number of chromosomes as the parent, whereas in meiosis, that number is reduced by half. Now again, it is important for you to start trying to track the stages through. The nice thing about mitosis and meiosis is that the stage or the phase names are the same. And the difference with meiosis is that we go through the steps twice. Importantly, this is not a cycle. This is not part of the normal cell cycle, which we talked about in mitosis. This is a separate process, and it's pretty much an open loop. It doesn't circle back on itself. And that's important. It's important to be aware of the fact that these cells are not just going to keep going round and round the loop. There's a specific purpose for the process of meiosis. And the other thing that's really important that we will have a look at during the modeling phase is this idea of crossing over. And this is another way in which we increase variation. Very briefly, crossing over involves exchange of genetic material between chromosomes. So it's therefore possible to have not identical chromosomes coming through into each of the ultimate gametes. There's some recombinations that are actually happening as genetic material is being switched from one chromosome to another. That's a very important part of the modeling process, and I think it actually belongs where we look at the modeling because it's much easier to see when you're modeling it than to talk about it. But you can see as we go through this cycle, everything kind of starts similarly to a mitotic division, but then the separation happens and we go through a second division without the replication that we had before. So the important things about meiotic divisions is we have a double division process, but only one replication stage, and we have this very important addition of crossing over that occurs. Now, when we talk about modeling, we'll also talk about some phenomena such as random segregation and independent assortment, and these are terms which we'll introduce in later videos, but they are also ways in which the process of meiosis introduces additional variation into the species, and therefore it's very important for the continuity of the species and the ability of the species to survive and adapt to different types of environmental changes that may occur. So these are the keys to this, and I think it's very important to go through the modeling process in order to see this. So that's something that you will do in class, and perhaps it might be something that I'll put together in a later video, just if her time permits me to do that. So here is a comparison of the difference between oogenesis, which is the process of producing oocytes or eggs, and spermatogenesis, which hopefully, obviously, is the process for producing sperm. The important things are that when fertilization occurs, the bulk of the material in the zygote, the fertilized egg, is actually going to come from the egg itself. So the egg is large, has a lot of cytoplasm, has a lot of cell contents, as well as the genetic material that's going to be important for the new developing individual. Sperm, on the other hand, basically just have genetic material they're going to pass on. Inside of the cell is going to be some mitochondria, because that's going to be the way of generating energy to keep the tail beating, and also some acrosomal vacuoles, places that will store some important enzymes and other chemicals that are needed when the sperm reaches the egg to penetrate through the egg and release its genetic material. So what we see as a result is some slight differences in the formation of the gametes. And you can see one of the things that we've tried to show is this kind of offset spindle. So when the spindle forms during oogenesis, it's asymmetrical. Which just means that it's actually kind of shifting across to the periphery rather than being perfectly in the center. And what this does is it produces, in that first division, two different sized daughter cells. So the smaller one is actually going to end up becoming two of the three polar bodies that are produced as a result of this process, whilst the larger one will develop into the egg and a third polar body. So you can see in the process of oogenesis, we get 4 cells, but 3 of those are little tiny cells with not much material inside of them, and the 4th is a much larger cell that is going to ultimately be the egg or the oocyte. In spermatogenesis, we have these tiny little cells. All 4 cells are the same size. They contain equivalent genetic material, not identical. Of course, the process of meiosis does not produce identical cells because they have only half the information as the parent cells, and because of that, they won't have identical information to each other, and because of things like crossing over, there may be additional reasons why there are significant differences in the genetic material in each of those cells. It's probably also worth pointing out at this stage too that after the cells have been produced in a meiosis for sperm, the specialization or the differentiation that occurs to those cells is a post-meiotic process where the flagellar tail will develop and the cell contents will be reduced to those things that I talked about earlier on. So we can see that this is an important process for reducing the number of chromosomes for the process of fertilization to restore that number once that occurs. But it's also a slightly different process whether we're talking about the formation of eggs in the female or sperm in the male. I did want to quickly touch on the importance of models. So this is something that obviously if you're looking at meiosis, you can stop here. But if you're looking at modeling, there's a couple of things that I would add to this, because modeling of mitosis and meiosis are the specific contexts for our study of these two processes. So it's important that we understand something about modeling in science. And I think the first thing is to give us reasons why we use models. So we use models to make predictions. The primary things we model are the very small and the very large in science. So we model the solar system, we model the universe because we can't go out and look at it. We also model chemicals, chemical substances, atoms, molecules, cells, because they're very small, they're very hard for us to observe. Of course, we're getting better. Our technology is getting better, so we're able to see things much smaller in more detail than we used to. But that's the main reason that we model things in science. So in a general sense, we model the very large and the very small. We use it to make predictions. Our better models make better predictions, basically. So as we improve our models, our predictions become more accurate, and then we can be more confident in the models that we have. Models also provide explanations in the same way that we have theories that help us to explain observed phenomena. What our models should help us do is to apply those theories to be able to understand exactly what's going on in any situation. And models also provide levels of consistency across different contexts. So, for example, when we talk about the model of the atom, we don't talk about the model of hydrogen or the model of carbon or the model of oxygen. We talk about the model of an atom as a consistent thing with maybe a few slight modifications, such as the number of protons, neutrons, or electrons, in order to explain differences in individuals. The same is true with cells. We've talked about animal cells, fungal cells, plant cells, prokaryotic versus eukaryotic cells. And in each of these cases, we're talking about some pretty common consistencies across all of those cells, with some specifics differing in each of the different groups. Models can come in a couple of different types. We can have simplified models, and most of our models are going to be simplified to a certain extent. We did build models of the fluid mosaic cell membrane earlier in the year, and we did that out of cake and lollies, and that could be regarded as an oversimplification of what's going on. And most models are static as well, so they're not actually moving and behaving in the same sort of way that active living models would behave. So sometimes our models can be oversimplified, and it's something to think about when you're using them. They can be mental models. Models is something that is probably well known amongst some physics concepts as well, in terms of trying to think about what sort of would happen in certain situations. But we do have mental models in biology as well, where we think about how things are structured together, how they might work, particularly how material might be broken down, exchanged, reconstructed when we're talking about processes that are too small for us to observe. Sometimes these models are express models too, and it may well be that you are looking at something like plasticine, or Play-Doh models or paper models, when we're looking at DNA structure and replication, and for chromosomes to represent the processes of mitosis or meiosis. And you can actually see these. You can draw diagrams of them. You take pictures of them. You can even do stock frame animations to show how each of these steps is occurring, and then sort of run them together to try and get a sense of the motion of the whole process together. And then some of our models are consensus models. So sometimes we talk about these. Good science is always about being tested by your peers, by having discussions in the scientific community, the biological community, and saying your model is robust, it stands up, it's got good explanatory power, it provides explanations for observed phenomena, or it doesn't, or there's weaknesses, or there's a limitation to the models. And this is good science, being able to discuss the effectiveness of our models, how useful they are, and perhaps how they can be improved is how science continues to move forward. So good luck with your building models, and keep in mind some of these important aspects of why we do model in science. Thanks for watching.